Hello, my name is Dr. Altura. I received my BS, MS, and PhD degrees from the UCLA School of Engineering and Applied Science. I've been involved in research for over 40 years. My areas are especially electrochemistry, chemical kinetics, and material science. I'm going to present an abbreviated video of what I have in a video that takes about 25 to 30 minutes. I would like for you to look at the overall video to appreciate what's going on over here. We're going to look at basically at Darwin's tree of life. Darwin proposed back in 1859 that life, that forms that we have today, are totally by chance, totally random processes. This is what's called evolution. It involves little steps over a lot of little steps over a long period of time. He formulated this tree. The tree consists of a single ancestor cell at the bottom of the tree, and the tree then progresses out to produce all the species we have today, millions of different species. What we're going to look at quickly, and you can see it in the other video, is we're going to deal with just a very simple model of Darwin's tree. We're not going to deal with the whole tree. We're just going to deal with the single cell going to the human being. But the human being is also very complicated. And we're going to simplify it, because that's, a lot of times I learned that even in my freshman year at UCLA, if you take a complex system and you simplify it, you can get a lot of information which you can then apply to the complex system. We're not going to deal with the whole human cell, the whole human being, because the human being is composed of 37 trillion cells. That's too large. We're also not going to deal with even a single human cell, because each human cell has over 20,000 different proteins which have different jobs. We're just going to deal with two proteins, BRCA1 and BRCA2. We know these are tumor suppressors. If they're mutated, this causes cancer. Ovarian cancer, breast cancer, etc. These proteins are very specialized. What do I mean by that? They have what's called an immobilizer function. Just like a car immobilizer, the proteins have to have the exact right combination so that you have both a geometric component, they fold into the right position, and an electronic component. Like a key that you use in an immobilizer has an little electronic circuit in the key and the geometry of the key plugging into the steering wheel. These two proteins are very complex. There's 1,863 amino acids in one, and 3,418 amino acids in the other. Again, a protein is a combination of amino acids. Their function is tumor suppression, and that's why, in this case here, for the single cell, we're not going to get into how we got to this, but we're using a model which is based on a bacteria called M. genitalium in the Minimum General Project, which was brought down to 382 proteins. Below this, the cell doesn't function. And we know, because this is a simple cell, and this cell does not have a problem with cancer, that these two proteins are not included in a single cell. Now, so we see that very complicated. The string has to be exact. They're multifunctional. They're involved in detection of mutations in the DNA. They're involved in repair of double strand, repair in DNA. They're involved in stopping the cell cycle so that the cell doesn't reproduce. And they're also involved in apoptosis, which means that the cell is sacrificed, killed off, if the mutations can't be fixed. They're totally unique. There's nothing in the human cell, other proteins which are similar to BRCA or BRCA1 or BRCA2, which means that they could not have been formed by piling together portions of these other proteins. And most important is the Goldilocks syndrome. That you have to, just like Goldilocks, the porridge has to be just right. There's only one combination for both of these that will work to make this a tumor suppressor. If just one amino acid is out of place, is missing, or there's one additional amino acid, these protein suppressors will malfunction either partially or completely and lead to increased rates of cancer. Now, as an example, if these are mutated, they already found for BRCA1, they found 1,700 different mutations. And of these, 850 of these are clinically significant. In other words, they cause cancer. So how many, we only have one unique combination that works for each one of these. Just one. How many different combinations do we have? So we have 5 types. We're just going to take an example of a necklace with one around the neck, 5 different types of pearls, 4 different positions. 1, 2, 3, 4 positions. Each one can have 5 different kinds of pearls. The number of combinations is 5 times 5 times 5 times 5, that's 625. The general formula for the combinations is the number of the types of pearls, 5 different types, and the number of positions, 1, 2, 3, 4. It's 5 to the 4th power, which is 625. Proteins are formed again from amino acids. Amino acids come in 20 different types for life. There are actually 40 different types because each amino acid also has a mirror image, but for some reason which we don't know why, in life only the left-handed amino acids are effective. So we have 20 different types. So for BRCA1, if we use this formula, we have 20 types, T, to the power of the different, how long the chain is, 1863, 20 to the 1863 power, or BRCA2, 20 to the 3418 power. These numbers are so large that we can't even conceive of them. Just this number here, with 3418 zeros, if I was to pronounce each zero, taking a half a second for each zero, it would take me more than a half an hour to pronounce all the zeros. If you compare the number of combinations here to the number of combinations of Powerball, Powerball only has 10 to the 9th combinations. But let's take something much larger, the observable universe. With all the trillions of galaxies and the billions of, hundreds of billions of stars in each galaxy, the total number of atoms in the universe is estimated to be 10 to the 80th. And the total number of particles in the universe only 10 to the 86th. Compare these again to the different combinations you can get for BRCA1 and BRCA2. This is impossibly, that one combination is impossibly hard to come by. Can you imagine trying to find, just for one, for example, the number of particles, trying to find one particular neutron in the whole universe. You're flying around at the speed of light, you want to find out one neutron. You can't find it, because it would take a very, very long time. You're talking about a universe that's billions of years wide, billions of light years wide. And you're traveling at the speed of light. It would be impossible even for just to find one neutron in the universe. And to find the right combination in these large numbers is basically impossible. It would take billions and billions of generations that you would need for this tree. And it would take billions and billions and billions times billions of years. Much more time than that the Earth has existed and that the whole universe has existed. If we take a look at the different paths from the single cell, which doesn't have these two complicated proteins, to the human being cell, which does have them, path is not important. It doesn't matter which way you go in the tree. The odds are all the same. And I'll give you an example. Powerball, which consists of 2 machines, white and red, has the same odds as if you generated these numbers with a PC, these random numbers. And if you're suspicious because someone hacked the PC, you can then use a roulette wheel. Well, you have 2 roulette wheels. One roulette wheel is white with 69 numbers, and one roulette wheel is red with only 26 numbers. The numbers that Joe Smith got to win the Powerball, the odds are the same whether they use the Powerball machines, whether they use the PC, or they use the roulette wheels. And this is true over here also. No matter which way you go, from a single cell where these two are missing to these complicated proteins and the genes that code for them in the human being, the odds are held the same. It doesn't matter how you get there. what kind of evolution you use, what kind of selection you use, etc., the odds still remain the same. And the odds are impossibly small. It would take many, many, many more billions and times billions of years to get just these two proteins into the human genome and the human cell. And the same thing goes for all average proteins, because an average protein in the human being has 300 to 400 amino acids. That means 20 to the 300 or 400th power, which is still much higher than these two. And then we can go to the titin, which is the largest protein in the human body, which has, believe it or not, 34,000 amino acids, which have to all be in the right combination. One is out of place. This can cause muscular diseases, because titin is used in the muscle to give the springiness of the muscle. So this is not just a game we're playing here. This is not just an intellectual pursuit between intelligent design and evolution. This has a very practical significance here, because we're talking about saving many, many thousands of human lives. The perspective you have is important. I've learned that over 45 years of being involved in research. So to get a solution to this problem and to save thousands and thousands of people's lives, we should have the right perspective. And basically, the only way this could happen by chance is if you had billions times billions of years, which you don't have. Thank you. Please watch the larger video. It's available on YouTube. It's available on my channel to see a more intensive discussion about these two proteins. Thank you very much.